Hey, hello everyone. My name's Dr. Andrew Mason. I'm a lecturer at the University of York and an Elixir Data Stewardship Fellow. So today I'm going to walk you through the exercises for the ENTHUSE bioinformatics practical about P53 mutations in cancer. So the first thing I've done is I've loaded up the Cancer BioPortal, so this homepage here, cBioPortal.org. The first thing to notice is there is a huge list of available studies. So this website has a massive amount of publicly available cancer sequencing data, looking at things like mutations to understand different types of cancer. So each of these studies is published work with data available for the entire community to access. This is a public website, so anyone can do this, not just cancer researchers. So different size samples for each of these different cohorts. And today we're going to be working with the Cancer Genome Atlas, TCGA, the Pan-Cancer Atlas. So if we select this here, we've loaded up 32 different cancer studies, leading to almost 11,000 different samples. So the Cancer Genome Atlas is a massive international effort looking at a whole range of different types of cancer. It's a really great resource for understanding TP53 mutations across different types of cancer. So let's query here. We'll put in TP53, it's a recognized gene symbol, we submit our query. So this now is going to load up all of these different samples so that we can start understanding what P53 mutations are doing in cancer. Now, this is called an oncoprint. Now, I know it's very colorful. There's a lot of information here because we're now looking at almost 11,000 samples. The key thing to look at here is that it's P53, TP53, the gene name, with all these mutations. So 36% of those 11,000 samples have some kind of mutation in this gene. Now, if we zoom out, so we go to 1% here, then we can see that each column is a different cancer sample. So you can create blocks, you can look at particular ones, you can hover to get information on these different tumors, or you can use the slider. So you can already see here that there are different colors for different types of mutations. So the ones that you'll have come across in A-level biology before, in black here, so these are nonsense mutations, so truncating. So they create a stop codon and they shorten the protein length. In green here, we have missense mutations. So these are ones where the amino acid changes as a result of the mutation. One that people maybe haven't come across at A-level yet is these ones in blue here. So these are copy number changes, blue and red as well. I'll just zoom in on that bit. So you can now see quite clearly that each column here is a different tumor. So you can see this one here, part of this study. So in blue, these are deletions. So instead of being diploid, so having two copies of the genes, in these cases, there are no copies of P53. And that's because the genome is quite unstable in cancer, and so sometimes you get deletions. And that can be really important for tumor biology. These ones in red, these are amplifications. So you're getting more than the two copies that you would usually expect. Okay, so this gives us full cohort-wide. So let's look at where those mutations actually are. So we select the mutations tab at the top here, and this gives us what we call a lollipop plot. Okay, so a lot of data, that's why it's a little bit slow to load. So if we look at the lollipop plot here, what we're showing is the length of the protein along the x-axis with the different important domains in green, red, and blue. So these are the functional bits of the protein that actually do its job. So in red, this is the DNA binding domain. In blue, this is where P53 binds other P53 proteins so it can do its job. The lollipop bit comes from these stick and ball parts of the plot. So this is effectively showing how common a mutation is. So the higher the stick, the more common a mutation is. Now this R273 for H, but other mutations at the same site. This is a hotspot. It's the most common amino acid to change in P53 across all these cancers. And this is the mutation that we looked at in the wet lab practical when you came to the University of York. Now we can see here we've got black, which are the truncating, and we've got green. So this reveals a bit of a different function. So what we'll do here is we'll select down and we'll just look at the truncating mutations. Now you can see that these truncating mutations are located all the way across P53, perhaps less so right at the end. And the reason there is that a truncating mutation breaks the protein, stops it doing its job. So these can happen all over the protein, but if it's right at the end, usually it's less likely that it has a negative impact. Now, if instead we focus on the missense mutations, so these are the ones where you change the amino acid and perhaps change the functionality, you can see that all of these mutations are actually found in the functional domains only. So including our one we looked at in the wet lab practical, by changing the amino acid chemistry, we're changing what the protein can do. Okay. So the next thing to look at is whether or not changes in P53 change survival status. So if you have this mutation, do you have a worse prognosis? Are you less likely to survive than people without the mutation? So we can look here at this comparison survival tab and go straight to the survival subtab here. And this is what we call a Kaplan-Meier plot. Effectively, we're plotting the percentage of the cohort with a mutation here that are still alive over time. And you can see in red here, this is where the samples have a P53 mutation. You can see that the survival is much worse if you have a P53 mutation compared to if you don't. Now, one of the really important things here, that one of the exercises in the tutorial is to think about what could be confounding this work. So with biology is noisy data. There's always a lot of personal variation in data. And this is also covering lots of different cancers. So different cancers may have a different response to P53. It may be that it's present in different frequencies across different cancer types. There might be differences in sex that are completely hidden here, differences in how well different cancers are treated, and so what survival differences we're getting, the different severity of different P53 mutations. It's all hidden here. These are all things that we have to consider and perhaps control for when we're looking at cancers and trying to understand the effect of the mutation. So that point on P53 mutations across different types of cancer, we can look and see if it's the same or whether it's very variable. So we go to the cancer type summary here, and you can immediately see these are the 32 different studies of TCGA. You can immediately see that the frequency of mutations changes a lot. So in these uterine carcinosarcoma or in esophageal cancers, the frequency is very high. Over 80% of patients have a P53 mutation. Then you go all the way to the other end of the spectrum, something like acute myeloid leukemia, AML. It's down at less than 10%. And in some cases, there are very, very few mutations at all. So you can split this further by different types of cancer, going all the way down, not just at the study, but going right down to the details of the different types of cancer that are in each study type. It's also good to remember that these different cohorts are different sizes. So if you change it to counts, it changes the order. So now you've got, just because the cohorts are bigger, so the number of samples that we used in the project, lung cancer, ovarian cancer, head and neck, breast cancer, colorectal, they're the big players in this cancer project. So there's lots more of them. Okay, so what we're going to do now is use this distribution of different types of cancer to explore some case studies. Now, the first one I want to explore is the one that I would say looks the most different in this bar chart. So most of these bars are green, and they're just missense or truncating mutations. This one here, the sarcoma dataset, you can see there's lots of blue. Now these blue are deep deletions, so that copy number change that I mentioned before. So instead of just breaking P53, we're actually removing P53 entirely. So we can hover over this bar here and query the study for P53. So in Chrome, this opens a new tab, so it's quite easy to study here. Okay, so we're back to the oncoprint, and you can see that 47% of the of the cancers have a p53 mutation, so this is higher than the average. But if we go to the survival plot again, you can see there's actually no difference in survival. So across all cancers, there was a difference in survival, but not within the sarcoma alone. If you look at the mutations plot, you can see that whilst there are fewer mutations overall, just because of the size of the study, most of the mutations are happening in this DNA binding domain, as we'd suggested before. So there's, we wouldn't expect there to be major differences. So why isn't there a significant difference in survival? Well, in biology, proteins very rarely act on their own. They work together in pathways to do functions. So let's look at pathways. This is effectively lots of different proteins working together. Now, this pathway that comes up straight away is the p53 pathway. So you can see p53 in its usual activity takes cells towards senescence. This is the aging factor, and apoptosis, that's cell death. And p53 activity stops proliferation and growth. So you can see it's a stop because it's an arrow with a flat head, so it inhibits proliferation. So what we're seeing here is the percentage of mutations, that 47% that we saw before. But you can see that other genes in the pathway are also heavily mutated. So MDM2 here inhibits p53, but is very mutated. And CDKN2A inhibits both MDM2 and p53. So there's a lot of mutations here. And it would be weird if we want to get rid of p53, why would you also want to get rid of a protein that is blocking p53? So what's going on here? Is that actually what's going on? Well, instead of just looking at p53, let's look at all three of these genes. So we add to here MDM2 and CDKN2A. So these are the two genes that we are looking at in this plot. Submit this query. So we've loaded up to the pathway. Let's look at the oncoprint first. First thing to notice actually is that now 74% of the patients have a mutation in at least one of these genes. So it can't all be happening that people have mutations in all three genes at the same time. And sure enough, that's what we see with this oncoprint plot here. This top row here, p53, set of mutations, MDM2 mutations actually only happen if there is not a p53 mutation. CDKN2A is a bit different. There's a block here, and actually there's a lot of deletions when it's a change of function to the missense mutations from before. So this is what we call mutual exclusivity. So if there's a mutation in one, p53, then there isn't a mutation in the MDM2. Now, interestingly, the mutations that are in MDM2 are amplifications. So this is making more MDM2. So we predicted from the pathways that it would be weird if MDM2 was deleted when actually it inhibits p53, and that's what the cancers want to inhibit. But actually what's happened is there's amplifications of MDM2, and so that means that there's more MDM2, more of an inhibitory effect on p53. So actually, what's important here is that we're mutating lots of different parts of the p53 pathway. So instead of just hitting p53, we're hitting p53 and the things around it. So let's see if that has had any impact on the survival analysis. And sure enough, if you have a mutation in at least one of these genes, your survival is much, much worse. Whereas if it was p53 alone, that survival was like the group that didn't have any of these alterations. So it's important for subtyping that we look at not just one gene in isolation, but the genes that it works with in its pathway. So another set here that's quite different. We're going to go down to where TP53 is not very common. And we're going to look right down here at AML. So select the study again, query the study for p53. So only 9% of patients have a mutation in p53. And because of that, there's not really any evidence of a hotspot at all. Mutations are in the places that we would kind of expect, but there's not one site that's being particularly selected for. And interestingly, though, despite the fact that it's much lower numbers, p53 mutations are really, really bad for survival. So AML is a blood cancer, and as a result, they don't actually have that many mutations. But one other gene where there's a very well-characterized hotspot is a gene called NPM1. So we're going to add this to the query and have a look at it here. Now, straight away, coming back into the survival, you can see that it's still significantly different, but interestingly, they have very different survival characteristics. So P53 is much worse. NPM1 is also worse than the unaltered, but not as bad as P53. Now, interestingly, they're significantly mutually exclusive, so they're not happening at the same time. We can plot that nicely here in the oncoprint. So if you have a mutation in NPM1, you don't have a mutation in P53. Now, these two genes are not in the same pathway. Just like in the example we just did with the sarcoma, they're not working cooperatively. It's a very, very different type of tumor that's happening here. So this is actually more of a choice. Imagine a ball rolling down a hill. It could go down one side of the hill or another if it's rolling down into valleys. So the idea here is that if you have a mutation in P53, it takes you down a certain trajectory. If it's an NPM1, it takes you down a different trajectory. So these are very different subtypes of the cancer. So these would require different treatments. And this is all about personalized medicine. So using public data from the clinic to understand the cancer better, and then work on ways to provide the best treatment. And the final case study I'm going to talk about is this head and neck squamous cell carcinoma here. So mutated in 68% of the cases, so mutated in a lot of the tumors. And I'd encourage you to look through all these different plots to understand the differences, particularly as the survival is worse in the P53 group, but not that much worse. But one thing I want to show you here is actually to look at the clinical data. So this is lots of information that's come from the clinic. And the key thing here is the fact that the subtype of head and neck squamous cell carcinoma is significantly different between whether you have a P53 mutation or not. Now in this bar chart here, you can see that in the altered group, it's almost all purple. So these are a particular subtype of head and neck squamous cell carcinoma. And in the unaltered groups, so those without a P53 mutation, there's this big green chunk here. Now this is because in head and neck cancers, they're caused either by smoking or by human papillomavirus, HPV, the same virus that causes a cervical cancer. And you can see in the unaltered group, they are enriched for being positive for HPV, and the altered group has very few HPV positive tumors. So again, just like with the AML example, this represents potentially two very different types of cancer biology going on here. So what I want to do now is investigate this a bit further. What are the differences between these subtypes? Because if it's just that HPV is really bad or really good for the tumors, it could be that P53 isn't the important thing at all. So we can select on the project up at the top. It takes us to this enormous dashboard showing off really just how much data there is on the cBioPortal. Now, if we go down here to the subtype pie chart, we select HPV negative and HPV positive tumors and compare them, we can see that in terms of the survival, the HPV positive tumors, the ones that are typically negative for P53 mutations, have got better survival. So it is a confounding factor that the HPV positive tumors are the ones that typically just have better survival rather than the P53 dragging the survival down in lots of these cases. It's quite a tricky thing to unpick. Now, because of the power of these studies, we've got access to a whole load of data. So I've just clicked on the genomic alterations here. So what mutations are enriched in the HPV positive and the HPV negative. And you can see P53 coming up in the HPV negative group here, just like with CDKN2A. So that same pathway activator. It's not until we get down to TRAF3 here that we see an enrichment in the HPV positive group. Now, interestingly, TRAF3 gets deleted in these head and neck cancers, and TRAF3 actually helps to recruit antiviral factors. So by deleting it, it helps the HPV cause the cancer. So it's one of the amazing things really with the Cancer BioPortal is we have all this information at our fingertips. We're able to research new questions. We can use existing data, so we're not having to get more patient samples. We can reuse data. We can share data with the community. It's a fantastic resource. I hope this walkthrough has been informative, and good luck with the questions on the website.